Northern blotting is for RNA, southern blotting is for DNA, western blotting proteins, eastern blotting post-translational modification, to identify post-translational modification. Bioluminescence is displayed by which animals? Dinoflagellates. Okay, 152. Which of the following causes an increase in sodium reabsorption in the distal convoluted tubule? Increase in aldosterone levels, increase in ADH levels, decrease in aldosterone levels, decrease in ADH levels. Aldosterone causes what? In the blood pressure regulation mechanism, in the regulation of blood pressure, what is the role of aldosterone? Aldosterone contributes to increase of blood pressure or decrease of blood pressure? Increase of blood pressure. By what mechanism? By increasing sodium reabsorption. By increasing sodium reabsorption. Give me a counter hormone to aldosterone. ANF, anti-natriuretic factor or anti-natriuretic, atrial natriuretic peptide, cardiac hormone. Okay. ADH influence on blood pressure. What is the influence of ADH on blood pressure? Reduces, sir? Increases? Increases? Why do you say so? Huh? Its another name is vasopressin. Even if you don't know the mechanism of how ADH acts on the blood vessels, it has a name, no? It has another name. Vasopressin. Vaso refers to blood vessels. Pressin is to compress. If you reduce the diameter of the blood vessel, what will happen to the pressure of flow? Increases. If you increase the diameter of the blood vessel, what happens to the blood pressure? Decreases. If you increase blood volume, what happens to blood pressure? If you increase blood volume, what happens to blood pressure? Decreases, sir? Increases. Why? Blood pressure is the pressure required to keep blood in circulation. That's all. Don't blindly apply the inverse relationship of pressure and volume. Right? If there is increase in volume, pressure will decrease. If there is decrease in volume, pressure will increase. That is for the tube. Pressure-volume relationship. What is that law? What law is that? Pressure-volume relationship? Huh? Boyle's law. Boyle's law is it applicable for fluid or is it applicable for the tubes? If you increase the volume of fluid, the pressure exerted by that fluid on the surface will be more or less? More, no? So you should apply common sense. If you increase the volume of the tube in which the fluid has to pass, what will happen to the pressure of flow? Pressure will decrease. It is just like you are using a pipe to water your plants. Water is flowing at a particular pressure. If you want to water a plant that is far away, what do? You will block the mouth of the pipe, opening of the pipe. If you narrow the opening of the pipe, what will happen to pressure? Increases and the water is thrown for a further distance. Common sense. No, you have done this experiment. You have done this activity. Whether you have looked at it as an experiment of pressure and volume relationship is a different issue. But this activity all of you have done. Correct? How do you do that to pour water on your friends? You have the pipe. Wash car, wash your vehicles. Not do that, no. Okay. What will happen to volume, or what will happen to the blood pressure if the elasticity of the blood vessel is increased? What will happen to blood pressure if the elasticity of the blood vessel is increased? These are the three parameters which will influence the blood pressure. elasticity of the blood vessel, diameter of the blood vessel, volume of blood. Two parameters related to the blood vessel and one parameter related to blood itself. So increasing the volume or the diameter of the blood vessel decreases pressure. Decreasing the diameter of the blood vessel increases blood pressure. That is clear. An elastic vessel versus a rigid vessel, or more elastic vessel versus a less elastic vessel. Pressure will be more in which of the two? More the elasticity, more the pressure required to push blood. Is it? Elasticity is the ability of a substance to regain its original form when a deranging force is applied. Can I say that? When there is a force that is applied to derange the particular object, if it is elastic, it will apply counter pressure, it will exert counter pressure to get back to its original position or get back to its original shape. When the blood is flowing through the blood vessel, the blood is hitting on the wall of the blood vessel also. There is some pressure with which the blood hits the wall of the vessel. If the wall is elastic, what will it do? It will push back. When the wall is also pushing the blood back, will the blood flow more smoothly or will the blood flow become lesser? Will the blood flow reduces or will the blood flow sustains for longer? It sustains for longer. Otherwise, if this is rigid, when it hits it, it will absorb it. The surface, the blood vessel wall will absorb the pressure. So the pressure that you have applied to make the blood flow is lost when that pressure is used to hit the wall. So and there is the pressure reduces in this for the flow of this. So to ensure that the flow continues to happen, you have to create more pressure. And that is blood pressure. In blood pressure you should understand, it is not you should not look at it as the pressure of the flowing fluid. Pressure created by the flowing fluid. In blood pressure you should always look at it as the pressure required to keep this fluid flowing. You understand? Blood pressure is the pressure required to keep blood flowing in the blood vessels. What supports it will reduce blood pressure. What opposes it will increase blood pressure. Anything that creates resistance to the flow of blood will increase blood pressure. Anything that will exert lesser resistance will decrease blood pressure. So rigid tube offers more resistance or elastic tube offers more resistance. Rigid tube offers more resistance. Elastic tube offers lesser resistance because it is pushing the fluid back into the compartment. So it is contributing to maintaining the pressure. Rigid tube contributes to dissipation of pressure. That is why as a person ages, baseline blood pressure keeps increasing. For a 70-year-old person or a 65, 70-year-old person, 130 to 140 baseline blood pressure is normal. Because the elasticity has become lesser. The blood vessels have become more rigid. All right. If you want to increase blood pressure, you give vasoconstrictors. If you give vasoconstrictors, it will increase blood pressure. If you want to decrease blood pressure, you give vasodilators that will reduce blood pressure. You should get the logic behind blood pressure. If you just get this logic, it is resistance. Anything that will increase resistance increases blood pressure. Anything that will reduce resistance reduces blood pressure. If you look at it from that point of view, you can answer any question related to blood pressure. Just on this logic, I can give you 100 questions on blood pressure, all worded differently. So what is the point in going through 100 questions? Get the logic right. You will be able to answer all the 100 questions. Is that clear? So aldosterone is produced in which part of the body? Which part of the body is aldosterone produced? Huh? Aldosterone is produced in hypothalamus. Huh? Adrenal gland. What part of the adrenal gland? Adrenal cortex. Adrenal medulla produces two hormones. What are they? Adrenaline and noradrenaline. Catecholamines. Catecholamines are produced by adrenal medulla Adrenal cortex produces what hormones? Glucocorticoids, mineralocorticoids. In mineralocorticoids, you have aldosterone, very important one. Not just sodium reabsorption, aldosterone causes potassium excretion. It increases potassium excretion and increases sodium reabsorption. ADH we discussed. Okay. What is the system in which aldosterone is also a part? A system which regulates blood pressure, in which kidney is at the center of it. Kidney produces one more hormone which regulates blood pressure. Which hormone starts with R? Renin. What is the system? Not the countercurrent mechanism. What is the system which is used for regulating blood pressure? Renin, angiotensin, aldosterone system. RAAS. Renin, angiotensin, aldosterone. All three contributes to increasing blood pressure. Renin will convert angiotensinogen into angiotensin. So angiotensin will bring about vasoconstriction. Angio is blood vessel, tensin is tension, constriction. Aldosterone brings about sodium reabsorption. See both of them together. One is causing constriction of blood vessels and the other one is causing sodium retention. Sodium retention, how does it help in increasing blood pressure? How is sodium retention or sodium reabsorption contribute to increasing blood pressure?
Sodium reabsorption make it hypertonic solution, so it will reabsorb more water.
Sodium is an osmotic substance. More sodium in blood means the osmolarity of blood increases. To maintain the osmolarity of blood, there is more water absorbed into blood. Volume of blood increases. So what will happen if there is more blood volume? Blood pressure increases. That's how it works. That is why if somebody is hypertensive, they are advised not to consume additional salt. And if they cannot not have additional salt, they are advised to consume rock salt. Why? Why rock salt? Rock salt has more potassium and less sodium. Rock salt has more potassium and less sodium. Alright? Can diabetes lead to hypertension? Yes. How? Diabetes is increased glucose in blood. Increased glucose in blood means what? Glucose is also osmotic substance. What will more glucose in the blood do? It will cause more water retention. More water retention, increased blood volume. Increased blood volume, increased blood pressure. Diabetes is more likely to cause hypertension than the reverse. Hypertension causing diabetes is very unlikely. But diabetes causing hypertension is very common. So somebody is diagnosed with diabetes first. If he does not manage his diabetes well, eventually he will develop hypertension also. But you will find many people with hypertension not diabetic. My father is a hypertensive, not a diabetic. Alright? If somebody has both diabetes and hypertension, you inquire with them what is it that they were diagnosed with first. You talk to them, ask them what was the first diagnosis. Were they first told that they were diabetic or hypertensive? Most of the times you will find that they were told that they were diabetic. It might be even so that they ignored it for a very long time and diabetes would have led to hypertension also, and hypertension was the first one to be identified. That's also possible. But it may not be diabetes detection or diabetes diagnosis at an early stage. It might be a later stage diagnosis. Diabetes could have been for a long time. All right. Everything is logical, students. Everything has logic. So you need to go behind understanding the logic. You need to find out why is this so? How is this doing this? Nothing is just like that. All right. So if you develop the approach of trying to figure out logic, see, it's not learning logic. It is, understanding logic. To learn logic and to understand logic, there is a difference. If I teach you the logic, that's also, you will still get it. But if you put your mind to figure out what the logic is, you might get it wrong. It might be wrong, you might not get it also. That's fine. But the exercise of trying to find out the logic behind the natural mechanisms will train your mind to look at things in a different way. When the teacher also gives you the logic, suddenly the missing links will get filled up in your brain. You have to create a path. Thinking creates a path. Not only a single path, linear path. If I teach you something, it is one path. One line. There is a problem, I told you the solution, I gave you the path, straight path. But you know the problem, you don't know the solution. You are searching for the solution. You are searching for the logic. You would have traversed multiple paths in the search for a solution. So all those paths, neural pathways will be there in your brain. Even after a lot of attempt, you don't get it. At the end of it, when you feel you are not able to get it anymore, you ask the teacher and the teacher gives you. There is the starting point, there is the end point. You would have gone in multiple directions. Now you get the end point, either by yourself or by the help of the teacher. Here to here, the path will automatically get drawn in the mind. The pathway gets established and the mind remembers it. It will be a powerful, strong pathway. And the best part is, all those different directions you had gone, from there also it gets connected. So this destination can be common to 100 different origins. All right. You cannot start with a single destination, start with the origin, reach the destination, build one path by the teacher's help, start one more, build another path, start one more, build another path. No. You will not have enough time in your entire lifetime to do that. You have to do chaotic exploration to figure out a solution. Multiple pathways gets crystallized. It's very powerful. Mind you should understand. You should understand the mind. Use the mind for your benefit. It's a tool. It's a powerful tool. It's like internet only. More powerful than the internet. You know how to use the internet, you have the world in your hand. Just like that. It's like a very cheap representation of mind. Mind is much, much more powerful. Okay. The difference you can consider like a plastic prototype of an iPhone. Looks exactly like that, but it doesn't work. Or you can only make sounds, twinkle, twinkle, little star, and whatever, Johnny, Johnny, yes, Papa. And the latest version of the iPhone. That's your mind. The plastic version, that too China made, is the internet. Imagine the power. All right, move next one. Select the correct matching of the type of joint with the example in the human skeleton. Cartilaginous joint between frontal and parietal bone. Is it right? Is cartilaginous joint between the frontal and parietal bone? Is this cartilaginous? What type of joint is that? Fibrous joint. Pivot joint between third and fourth cervical vertebrae. Is it right? Pivot is what? One axis around which the other bone moves, which is atlantoaxial joint between the first and second vertebrae. Atlas and axis joint. Atlas is the axis is the pivot and atlas moves around it. All right. That's why we get this movement. And because there is other range of movement also, this is also possible. Otherwise, we wouldn't have been able to do this. Okay. Hinge joint between humerus and pectoral girdle. Is it right? Hinge joint is elbow and knees. One plane movement. Only flexion extension. Gliding joint between carpals. Is it right? Yes. Gliding joint is correct. All right. We have already seen different types of joints and discussed it. So we'll move forward. Stimulation of a muscle fiber by a motor neuron occurs at neuromuscular junction, transverse tubules, myofibril, the sarcoplasmic reticulum. This is a straightforward, simple question. It happens at the neuromuscular junction. What is the neurotransmitter released at the neuromuscular junction during muscle contraction? What is the neurotransmitter released at the neuromuscular junction? Correct. Acetylcholine. Acetylcholine will go and bind to the acetylcholine receptors on the muscle fiber post junctional membrane, and that is what will trigger contraction. What should happen to acetylcholine after that? It has to be cleared. Who clears it? Who clears it? Acetylcholine esterase enzyme. There is a disease in which acetylcholine esterase is being removed. Autoimmune disorder. Myasthenia gravis. As it is, I am deaf. I can't hear properly. All self-made because of shouting for too long. And you cover your mouth and give me the challenge of identifying where the sound is coming from. And get to know what the answer is, what the response is in that muffled voice. At least have some mercy. All right. Okay. Myasthenia gravis. Remember, myasthenia gravis is neuromuscular junction disorder. Acetylcholine esterase is attacked by autoimmunity. Now acetylcholine esterase is attacked. Gone. What will happen to the muscle then? It will continue to contract. It will continue to contract. Involuntary contractions of the muscle, twitches of the muscle, even when you don't want it. Involuntary twitching of the muscle, myasthenia gravis. All right. Okay. Transverse tubules. T tubules are where? Where are the T tubules? In the sarcoplasmic reticulum. What are they responsible for? T tubules? Calcium. The sarcoplasmic reticulum in the T tubules, you store calcium. It helps in the muscle contraction, but that is not what is transmitting impulse. Myofibril. Myofibril is the contractile unit of the muscle. Individual fibrils. All right. Is it the muscle? Complete muscle cell? There are multiple myofibrils in the muscle cell. Sarcoplasmic reticulum stores calcium ions released during muscle contraction, not the site of stimulation. All right. Injury localized due to the hypothalamus would most likely disrupt. If hypothalamus is injured, what might get affected is the question. So you should know what is the function of hypothalamus. Let us see. Short-term memory. Is hypothalamus associated with short-term memory? What is associated with short-term memory, if any of you have come across? Cerebrum. Okay, cerebrum is a cerebellum. No. There is a region called hippocampus, like hippopotamus. Hippocampus. Hippocampus is associated with short-term memory. Okay. Coordination during locomotion. Is it associated with hypothalamus? Locomotory coordination is brought about by? That is brought about by cerebellum. Cerebellum is responsible for coordination. Executive function, such as decision making. Cerebrum, cerebral cortex, especially the prefrontal cortex. That is the decision making area. This is the area. All right. There have been experiments done. Experiments not by damaging the prefrontal cortex. Experiments done on people, studies done on people whose prefrontal cortex got damaged due to accidents. Now, without prefrontal cortex, this is a very important study. Without the prefrontal cortex, the person became more calm, more composed, more happy, more relaxed, less abusive, less judgmental, less jealous. This is all in comparison to his previous self only. What he was before prefrontal cortex damage and what he is after prefrontal cortex damage. Now, all the problems that humans are facing is because of higher intellectual ability. You have the ability to think, rationalize, develop logic, contextualize things, analyze things. Analysis leads to judgment. Correct? Aspirations lead to insecurities. Competition coupled with aspiration leads to jealousy. You also want the same thing that somebody else is wanting, and you know there is competition. Your mind is making you jealous about the person who is able to get it. So what do we have to do if we have to be happy? If you want to be happy, what should you do? Not destroy the prefrontal cortex. Learn to manage the prefrontal cortex. How? Yoga. Yes, there is no better human training tool, self-training tool than yoga. Nothing else works like that. Using your own body and your breath, you can take control of your mind. What better technology you have, you tell me? Every technology helps humans to become comfortable. Yoga is the only technology which helps humans to become powerful. Powerful as an individual, not powerful with tools or positions. All right. Anyways, I'm promoting yoga for good reasons. All right. Regulation of body temperature. There is thermoregulation center in the hypothalamus. There is hunger center in the hypothalamus. There is satiety center in the hypothalamus. All right. So these three are important centers that you will find in the hypothalamus. Temperature regulation, hunger, and satiety. All right. What is the hormone that is released by stomach, which will go and trigger the, not stomach, the blood. Adipose tissue cells will produce a hormone. You know that? Adipose tissue cells will produce a hormone. No? Which will go and tell the hypothalamus that I am already getting glucose, stop loading the body with more food. Satiety is triggered by a hormone produced by adipose tissue cells. We blame the adipose tissue cells for accumulating a lot of fat. But adipose tissue cells are telling your hypothalamus to stop eating. Before it can send that signal, we would have eaten a lot of food. Why do we tell that you have to chew your food? You have to drink your food. Meaning you have to liquefy the food completely in your mouth. How it works, I'll tell you. As sugar starts entering into blood, as glucose starts entering into the bloodstream, insulin is released. Insulin will do three things. This is also using your adipose tissue cells to reduce their load, to overcome your weight issues. As glucose starts entering into the bloodstream, insulin is released. Insulin will trigger three things. What are the three things? It will tell the cells to utilize glucose for their metabolism. It will tell the liver to convert glucose into glycogen and eventually fat, if glycogen stores are full. It will tell the adipose tissue cells to convert glucose to lipids, fats. So when glucose, it is in the order of priority. When cells are taking up glucose, they will take it up first. When cells have taken enough glucose, liver and adipose tissue cells are stimulated almost simultaneously. Adipose tissue cells, if they are starting to get glucose, it means that the cellular requirement is already met. The requirement of glucose for metabolism of the cells is already met. Adipose tissue cell will send a signal to the hypothalamus. It produces a hormone called leptin. This will not come in your exam. Leptin will not come in your exam, but you should know this. Leptin goes and stimulates the satiety center in the hypothalamus. When the satiety center is stimulated, that is when you start getting a feeling of fullness. You will feel satisfied with food. When we chew the food, what will happen? Carbohydrate digestion happens in the mouth. You should take small bite and you should chew your food for longer till it liquefies in your mouth. So even in the mouth, there is absorption of glucose. Glucose is entering your bloodstream from the mouth only. So glucose, as it keeps entering the bloodstream, before you overload your stomach with a lot of food, leptin starts getting produced because you are sending glucose through this only. Otherwise, what happens if you don't chew your food? Glucose is not entering the bloodstream through the mouth in adequate quantities to stimulate the adipose cells to produce leptin. In the stomach, there is no digestion of. So hypothalamus is the body's thermostat. Next question. Which one of the following statements is not correct? Anyways, deleted portion, so we'll quickly do it. Retina is the light absorbing portion of visual photopigments. Is it correct? It is correct. Retinal is the light absorbing portion of visual photopigment. Okay, retinal. In the retina, rods have the photopigment rhodopsin, while cones have 3 different photopigments. Yes or wrong? Yes or no? Yes. Rods are for night vision, cones are for color vision. Earlier, when I learnt it first, it was very confusing for me to remember rods with night vision and cones with daytime vision. In Kannada, my mother tongue, night is referred to as Ratri. Ratri means night. I remembered it as rods are for Ratri vision. Night vision. Ra. Easy to remember. Cones are for color vision. Color is during the daytime. Statement 3. Retinal is derived from vitamin C. Is it right? Retinal is derived from vitamin A. Rhodopsin is the purplish red protein present in rods only. Yes. So wrong statement is statement 3. Identify the hormone with its correct matching of source and function. Lot of questions from endocrine in this paper. Or at least the third or fourth question we are seeing from endocrine system. Oxytocin, posterior pituitary, growth and maintenance of mammary gland. Right or wrong? See, growth and maintenance of mammary gland with oxytocin, is it right? Wrong. Oxytocin does not contribute to growth of mammary gland. What contributes to the growth of mammary glands? Estrogen. Estrogen stimulates the growth of mammary glands. Progesterone helps in maturation of mammary glands. Continued stimulation of mammary glands with progesterone helps in maturation. Functionally, they become capable. So, it is during the first pregnancy that the mammary glands become functional. Until then, they have grown fully. Mammary glands require continuous stimulation of progesterone for maturation. Prolactin brings about milk production. Oxytocin only contributes to milk ejection. So, there are 4 hormones which are acting on mammary glands. Estrogen, during the entire menstrual cycle, continuously with cycle after cycle, with the stimulation from estrogen, mammary glands grow. And mammary gland growth, there are two components in the breast tissue, mammary glands and the fatty tissue. Mammary glands are equal in number in both males and females. There are about 20 tubules of mammary glands in each of the breast tissue. In females, the mammary glands grow and mature because of the influence of estrogen and progesterone. In males, there is no estrogen and progesterone stimulation, so mammary glands do not grow. But they are still there. There is no lesser or more number. Estrogen leads to fatty deposition in the body. And one of the primary areas of fatty deposition in the body because of the influence of estrogen is around the mammary glands. Which is the breast tissue. Alright. So functionality of the mammary glands and the fat deposition have no correlation. There is no correlation between the amount of fat in the breast tissue and the function of the mammary gland. Mammary glands will function irrespective of the amount of fat in the breast tissue. Alright. Melatonin is produced by pineal gland. Yes or no? Come on, quickly, we should be fast. Melatonin is produced by pineal gland. Yes. Regulates the normal rhythm of sleep-wake cycle. Yes, that is correct statement. Progesterone, corpus luteum, stimulation of growth and activities of female secondary sex organs. Wrong. Progesterone, corpus luteum is right, but its primary task is not growth and activities of secondary sex organs. Its role is to maintain the endometrium of uterus. Its role is to maintain the endometrium of uterus. ANF, atrial natriuretic factor, ventricular walls, increases the blood pressure. What is wrong in this? Ventricular wall is wrong. It is atrial wall. Increases blood pressure. Is it right? It decreases blood pressure. So both are wrong there. levels of melatonin increases as the day progresses and will peak in the night, or it is the highest in the daytime and decreases as the day progresses. What is right? Melatonin is the lowest in concentration in the morning and increases as the day progresses, peaking in the night, or is it reverse? Highest levels in the morning after rest and decreases as the day progresses and lowest when you fall asleep. What is right? Highest during? Meaning, increased levels of melatonin, does it cause wakefulness or sleepiness? Sleepiness. So that means at the end of the day, melatonin levels are higher. At the beginning of the day, melatonin is washed out. It is cleared from the circulation. Is it? Yes. Increased levels of melatonin is what will make you fall asleep. Temperature increases melatonin production. As the day progresses, because of the stimulation by temperature and light will increase melatonin production. What do I tell you to do during every break? Sleep for 15 minutes. You know what happens when you sleep for 15 minutes and when you drink water periodically? You are helping to keep your intracranial temperature low. With high focused activity, when you are studying, when you are learning, when you are doing mental activities, there is increased metabolism in the brain. What is the byproduct of metabolism? Heat. A lot of energy goes in producing heat during metabolism, wasted in the form of heat. So when you are studying, when you are doing a lot of intellectual activity, increased metabolism, this area, again, prefrontal cortex is the area of your body, is the part of your body which consumes highest glucose for its energy production. Very active area. So if you are continuously doing your studies, what happens? There is high metabolism in the brain, leading to increased heat, increased temperature in the intracranial compartment. Intracranial temperature rises, triggers melatonin production. 3 things will make you sleep: increased temperature, increased melatonin, increased carbon dioxide. Increased carbon dioxide is considered to be a consequence of increased activity. So sleepiness is a response to make you reduce your activity so that the demand for oxygen and the production of carbon dioxide is balanced. So carbon dioxide will be produced lesser when the activity goes down. So you are made to feel sleepy. Oxygen, temperature or carbon dioxide levels, increased temperature and increased melatonin. So when you have that, when you practice sleeping for 15 minutes of power nap, 15 minutes only, and you are constantly hydrating your body. With constant hydration, you are helping the brain to work optimally, because sluggish blood flow makes the organs get tired fast. Exchange of material becomes difficult. Oxygen supply is reduced. So drinking water maintains the blood flow. That is why you should not drink water when you are wait to feel thirsty and then drink water. Keep hydrating your body, 100 ml every 20 minutes. That's all. If you are awake for let's say your water drinking period is 12 hours during the day, 300 ml per hour over 12 hours, how much would it be? 3.5 liters. That is what many people tell you. You should drink about 3 to 4 liters of water every day. What many people do? Suddenly remember, so I have not had water. Gulp down half liter. It will only increase load on your kidney. It will make the blood more, diluted or more watery. Increases water. That will also cause problem. You drink too much water at a time, problem. 100 ml, 20 minutes. 100 ml every 20 minutes. These are all lifetime habits. All right, should practice. So when you sleep for 15 minutes, you are increasing oxygen supply. So whatever carbon dioxide accumulation has happened will get cleared off from the brain. You are allowing the brain to wipe out. When you sleep, melatonin, there is a mechanism that gets initiated which will start clearing melatonin. During wakeful state, melatonin keeps accumulating. When you sleep, melatonin washout happens. So you are reducing melatonin. during that 15 minutes, increasing your alertness. And what else do I tell you? Deep breathing. You get 5 minutes time, close your eyes, breathe deep. You're waiting for something to start, you're feeling restless, you're feeling anxious, you're feeling tired. That's all, sit straight. It increases energy flow. Close your eyes. Deep breathing, slow and deep breathing. Even 20 cycles of slow deep breathing. If you are doing slow deep breathing, you will be doing about 5 to 6 cycles in a minute, not more than that. So 5 cycles in a minute, for 20 cycles, it will take you 4 minutes. 4 minutes of slow deep breathing energizes your body. Add to that good food habits. You don't eat junk, you don't eat deep fried foods, you don't have sugar, you don't have damaging foods. See, you see that all powerful. The performance of this is dependent on a lot of things, which does not require too much training. See, anything that will add value to your life doesn't cost money, doesn't cost too much effort, it only costs decision. You have to decide, I'll do this, that's all, over. From that moment, you will start changing. Alright, okay. So, melatonin helped us to understand some strategies. When I create all of this as a part of the system, a lot of people ask me, oh, why is this? It's all very trivial things. Very trivial things. Why do you have to even insist on doing all of this? I will ask them, do you have a few hours to understand? No, we cannot bear you for 10 minutes. If you go on for hours, forget about improvement. I will be completely transported. So, just do it. You understand and do it, or you do it and understand, either ways you will benefit. So if you are not understanding and you have the misfortune of having to be stuck with me for some time, better do it, you will understand it. Alright. Fight or flight reactions cause activation of. What do they activate? Fight and flight reaction is a situation in which what gets activated? Sympathetic nervous system. What gets activated? The parathyroid glands, leading to increased metabolic rate. No, it is not parathyroid that gets activated. The kidney, leading to suppression of the renin-angiotensin-aldosterone pathway. No need for that. Or when the sympathetic stimulation is activated, when the sympathetic system is activated, what will be the influence of the sympathetic system on the renin-angiotensin-aldosterone system? Will it stimulate it or will it suppress it? Sympathetic system. Stimulated, because during hyperactivity, during a fight or flight response, you need to have good amount of blood pressure. You need to have the pressure to maintain rapid activity. So renin-angiotensin-aldosterone system is activated, not suppressed. The adrenal medulla, leading to increased secretion of epinephrine and norepinephrine. Is that right? That is correct. Those are the hormonal mediators of sympathetic response. Pancreas, leading to a reduction in the blood sugar levels. Is it so? Will pancreas be activated or not? Sympathetic stimulation, will it have influence on pancreas? Should it influence pancreas? Should it increase? Should it activate pancreas? If it is so, for what? For insulin. You want insulin to be produced during a stressful situation? Glucagon should be produced. You don't want compartmentalization of glucose. You want decompartmentalization of glucose. That means you want to bring glucose from compartments into bloodstream so that the hyperactive muscles get it. Organs which are working hard get it. You can manage that stressful situation, fight situation. Alright? When in a situation of fight, you are hungry. Let us assume you are hungry, very hungry. You are feeling very tired. You are on a trek. Next trip you will go for a trek. Let's say we will go to that mountain there. We are trekking. You are very hungry. Because of lack of budget, we have not given you any food. Alright? So tired, you are feeling like I cannot walk back to the bus. Suddenly a wolf starts coming towards you and chasing you. Let us not look at lions and tigers. Wolf. A wolf starts coming towards you. Will you tell the wolf, see, I am hungry, I cannot run, please wait? What will you do? What will happen instantly? Wow! Will start running. Where does that energy come from? Adrenaline and noradrenaline, epinephrine, norepinephrine. Suddenly it will kickstart. It is like a kickstart of the motor. Heart is stimulated. Heartbeat will start going to the roof. Breathing will match the heartbeat. Like a dog. Suddenly pancreas is stimulated. Glucagon is released. Glucagon will start breaking down glycogen, bring more glucose. Adrenal gland is stimulated. Glucocorticoids are released. Adrenal cortex, glucocorticoids are released. What will glucocorticoids do? Glucocorticoids also will bring in more glucose into the bloodstream. It will counter the influence of insulin. Glucocorticoids, if insulin is also being released, present there at in some levels, glucocorticoid will go and block the insulin action. It will not allow insulin to push glucose into compartment. So glucagon and glucocorticoids together, cortisol together will start pumping in more glucose into the bloodstream. In this situation, more than glucagon, it is cortisol, which will continuously keep pumping glucose into the bloodstream. You understand? So, sympathetic stimulation. So just think about the situation, a dog chasing you or a wolf chasing you, a robber chasing you, whatever, somebody chasing you. What happens is sympathetic stimulation. When you are in a yoga class, meditation, doing meditation, that is parasympathetic stimulation. All right. The shared terminal duct of the reproductive and urinary system in the human male is, what is the shared duct in the human male? Urethra, ureter, vas deferens, vasa efferentia. Urethra is the common duct. Urethra is common. Before that, what do we have? Vas deferens. You go higher up after vas deferens, as you keep going towards the testis, what you encounter? Epididymis. As you go further higher up, you encounter vas efferens. Then you get the rete testis. Then you go into the testis, you have the ducts there. Okay. What is surgically cut in male sterilization, surgical sterilization of male? Vas deferens. At what level? Vas deferens is present in the scrotum, vas deferens is present in the inguinal canal, vas deferens is going up to the testis. Where do you access that vas deferens and cut it? In the scrotum. It is just a 2 millimeter or a 3 millimeter incision. Pull the tube, cut it, ligate it, cut it and put it back, and one stitch. The person will not even know. The dressing will be like a band-aid on a small paper cut. But tubectomy is a major surgery. It's a major abdominal surgery. So if any couple wants to go for surgical birth control technique, surgical birth control method, you should motivate the husband to go for vasectomy, not tubectomy. Unless the woman is undergoing LSCS, lower segment cesarean section for the delivery of the baby, and tubectomy is also being done in the same surgery. You should not encourage surgery exclusively for tubectomy. Alright? It comes with its own risks. Vasectomy is easy. If the husband doesn't cooperate, give him a bottle of whiskey, put him down when he is drunk and drowsy, cut it off. Okay. Don't do that. It's all illegal. Without consent, any surgery is illegal. How much ever the wife requests you. Alright? The main function of the mammalian corpus luteum is to produce estrogen only, progesterone, human chorionic gonadotropin, relaxin only. Progesterone. Corpus luteum produces relaxin also, but the main function is not relaxin. Even without relaxin, delivery will happen. But without progesterone, pregnancy cannot continue. HCG is produced by whom? Who produces HCG? HCG is produced by developing placenta, we can say. Until the placenta is fully developed, the chorion of the developing placenta continues to produce HCG. Once the placenta is fully developed, there is no need to produce HCG. There is no need to keep the corpus luteum for progesterone production because placenta will continue to produce progesterone. Alright? If fertilization does not happen, what will happen to corpus luteum? What does it become? It degenerates, but what do you call it? What will be the color of corpus luteum when it is active? What will be the color? If you observe it? Luteum is a reference to the color only. Luteum is a reference to color. Corpus luteum, what color? Yellow color. It will be pale yellow in color when it is active. When it becomes inactive and is degenerating, it becomes whitish in color. So what will it become? Corpus albicans. Corpus albicans. Albino is a reference to pale. Something that has become pale, lost its color, is albino. Corpus luteum is when it is active. When it degenerates, it becomes corpus albicans. All right. So what are you testing for in a urine pregnancy test? What are you testing for in the urine pregnancy test? You're testing for HCG. You're testing for HCG. Tubectomy is a method of sterilization in which small part of the fallopian tube is removed or tied up. Ovaries are removed surgically. Small part of the vas deferens is removed or tied up. Uterus is removed surgically. Small part of the fallopian tube is removed or tied up. It is tied up and then removed. Both. It is not either or. Even when you remove it, you have to tie it up, tie the ends. To remove it also, you have to tie it and then remove it. Both ends. Alright. What do you call the removal of ovary? What do you call the removal? Tubectomy is removal of the fallopian tube or cutting the fallopian tube. Removing the ovary is? Hysterectomy is? Is it ovary? Hysterectomy is uterus. What is ovary removal called? Oophorectomy. Oophorectomy. Oophora is a reference to ovum. It means egg bag. Oophora is the egg bag. Oophorectomy is the removal of the egg bag. Salpinx is the reference to tube. Salpingectomy, Salphinx, salpinx is the reference to fallopian tube, is the technical term for fallopian tube. So if a question comes as salpingectomy, that is tubectomy only, fallopian tube removal only. Okay. Hysterectomy is removal of uterus. Which of the following is a hormone releasing IUD, intrauterine device? Multilode 375, LNG 20. LNG is what? LNG is levonorgestrel. It's a synthetic progesterone. LNG 20. Cervical cap and Walt. What are each of these? Multilode 375 is what? It is copper T. It is a copper based IUD. It's a copper based intrauterine device. It is not hormonal. LNG 20 is levonorgestrel. Levonorgestrel is a synthetic progesterone. It is a synthetic progesterone, that is a hormone based IUD. That's a hormone based IUD, levonorgestrel. Cervical cap is? Cervical cap is a barrier method placed on the cervix. You cover the cervix. If you put a cervical cap, is it permanent or temporary? Short term or long term? Short term. Why? Cervical cap will not allow semen or the sperms to enter the cervix and enter into the uterus. But on the other hand, it will not allow the menstrual flow also. It will not allow the shedding of the menstrual fluid, which would lead to complications. So cervical cap is not a long-term barrier. It's a barrier method. Like a condom or a vault or a cervical cap, these are all, or diaphragm, which is a female condom. All of these are barrier methods, not permanent ones. Assisted reproductive technology, IVF, involves transfer of ovum into the fallopian tube, zygote into the fallopian tube, zygote into the uterus, embryo with 16 blastomeres into the fallopian tube. See, in vitro fertilization, there are two techniques which you follow. This is an incomplete question. It is IVF ET or IVF ZT. IVF ET is in vitro fertilization, embryo transfer. That is a more commonly used procedure in which you don't transfer the zygote. You transfer the embryo higher up into the uterus, at the uterus and fallopian tube junction, and allow the embryo to slide and roll over. As it is rolling over, it will get implanted at the right spot. You don't put the embryo into the fallopian tube. You will put the embryo higher up into the uterus at the fundus, close to the junction of the uterus and the fallopian tube. That is IVF ET. IVF ZT is in vitro fertilization and zygote transfer. You can transfer zygote to the fallopian tube. You can transfer zygote to the fallopian tube. That is also there. But this here, it is giving both as the options. Embryo transfer as well as zygote transfer. And it mentions IVF. See, ovum into the fallopian tube is wrong. In vitro fertilization has already happened. Zygote into the fallopian tube is, that is correct, zygote intrafallopian transfer. It is called ZIFT, zygote intrafallopian transfer. But for that zygote also fertilization has happened in vitro. In vitro has happened, no, fertilization has happened in vitro. It is fertilization outside and transferring the zygote. Third one, zygote into the uterus. That is a wrong statement because zygote is not transferred to the uterus. Zygote is transferred into the fallopian tube. Embryo with 16 blastomeres into the fallopian tube. Embryo transfer is not done into the fallopian tube. Embryo transfer is done into the uterus. Now what do we choose as the right choice? None of the options are completely correct. All the options have fault. The first and second options are wrong. First and second options are wrong. Third and fourth options are partially correct. Both are associated with in vitro fertilization, but the location to which they are being transferred is mentioned as wrong. Which one do we consider? Option 3 or option 4? Now, if you have to make a judgment call, what will you make a call? Which one do you choose? Which one is more wrong so that you eliminate it? Fourth one is more wrong. Why? You don't transfer embryo to the fallopian tube. If you transfer 16-stage embryo into the fallopian tube, what will you do if implantation happens in the fallopian tube? It will lead to tubal pregnancy. Tubal pregnancy can lead to rupture of the tube and the products of conception going into the abdomen. It will become an emergency. It will become a life-threatening emergency. You don't transfer embryo into the fallopian tube. So that one is wrong. Though for third option, zygote being transferred into the uterus is also not completely right. We can still assume that it is being transferred at the mouth of the fallopian tube in the uterus. Zygote has to be transferred into the fallopian tube. So compared to the fourth option, third option is a better choice. Option 2 is right. Yeah, correct. Option 2 is right. Option 2 can be considered there. Alright, option 2 and 3, correct. Option 2 and between option 2 and 3, both are associated with IVF. Both zygote is produced through IVF techniques only, but transfer, our man, woman, they are children, we have to find out. This fellow's Father was colorblind. So XCY. Mother was normal. XX. So this fellow will not get the X chromosome from father. Both the X chromosomes of the mother is normal. So he will be normal, not colorblind. This lady, her mother was colorblind. Her father was normal. Father was normal. Mother was colorblind. She gets the normal X chromosome from the father, and since both the X chromosomes of the mother is colorblindness carrying genes, she is a carrier for colorblindness. Now, male children will get Y chromosome from the father. It can be either this X chromosome or this X chromosome. What is the percentage of male children likely to be colorblind? 1 of 2 is 50%. 50% of the male children are likely to be colorblind. Got it? You can solve it in your mind, but whenever you get percentages to be calculated in inheritance, don't take chance with mental calculation. Though it is easy to calculate in your mind, just write it down. Whatever symbols you use is according to you, but write it down and come to conclusion. All right? Don't take chance. It's okay if you don't get the difficult questions right, but you should not make a single mistake in a question which you could have definitely answered. All right? So those questions should not be lost. Those are your sure shot. Get the ones which you are supposed to get. What you are not supposed to get or what is difficult to get, it's okay. We'll see. Right? That is why level-based approach in dealing with the question paper. I hope all of you are practicing that. Running through the entire question paper to find out questions which you can answer in very quick time. Do that. Don't go from the first question to the last question, trying to answer everything. Your first priority is to pick up questions which are sure shot answers for you. Not a single of such questions should be missed out. They will save you time. Let's say if there are out of 180 questions that you are supposed to answer, so totally out of 200 questions, there are 60 or 70 questions like that which you can get it very easily. 70 questions, let us assume. So you complete these 70 questions in 45 minutes or 50 minutes. The kind of confidence your mind will get is very high. It will make you more focused and more calm. With more calmness and more focus, you are likely to solve even difficult questions. And then let's say out of the remaining 130 questions, about 60 questions require 1.5 minutes. So you are done with 130 questions, 90 minutes plus 45 minutes, 135 minutes over. 135 minutes over. You have answered 90 plus, 90 questions plus 70 questions. Not 90 questions, 60 plus 70. Even taking 1.5 minutes for 60 questions, you have still completed 130 questions in 135 minutes. The remaining questions are difficult ones, let us assume. You have another 65 questions left, of which you have to answer 45 questions. You have how many minutes? 200 minutes total. You still have 65 minutes. You still have 65 minutes and 45 questions to answer. Let us say out of the 45 questions you are supposed to answer, 25 questions are that which you cannot answer, you don't know. That means you have 20 questions and 65 minutes. You can still put in your time and solve those 20 questions. With 65 minutes, even if you can solve those 20 questions and you still leave out 25 questions, it's fine. So totally you would have attempted about 140, 150 questions. 150 questions, if you attempt, clearly, strategically, 150 is your 600. That's all, no? Gain advantage, strategy, it is strategic approach. All of you can do it. All right. Don't get bogged down. Don't have the unrealistic expectation of knowing everything. I should be familiar with everything. No need. 50 questions you did not know. It's okay. Out of the 200 questions, even if you are not able to answer 50 questions, you will still get 600. That's all. Don't get scared by the questions that you are not able to understand. Leave it. All right. Tackle all those which are easy for you first. First step. Tuck, 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 you should go. There will be many questions which you can answer in 20 seconds, 25 seconds, 30 seconds, 40 seconds, like that. When you cover all those questions in the first set of questions that you attempt, you will gain time. Use that time. Do the next set of questions which will require slightly around 1 minute to more than 1 minute to attempt it. Do it. Then go to the next ones which you are confident that you might be able to do it given more time. Do it. See, such questions are in physics only, no? None of the biology questions would be requiring 3 minutes for you to solve or 2 minutes for it. Either you look at it, you get it right away, 20 seconds, 30 seconds, 40 seconds. Or if you don't know, maybe you will have to think over it, you will have to build some logic. 1.5 minutes, done, over. At least a good number of chemistry questions will be like that. At least a part of physics questions will be like that. 50 plus 50, out of 100 questions in physics and chemistry put together, will you have 50 numericals? Difficult numericals? You don't have 1 in 2 question being numericals, no? In chemistry, about 25% of the questions will be numericals. Most of it is theory only. In physics, about 30-35% of the questions are numericals. Many are still theory only. So you have a margin of 50 questions, students. Don't get scared. Just stick to your process. Stick to your process, keep preparing, keep preparing, keep preparing. Retrospective learning. As you do this, you will become confident. And no matter how much preparation has happened, you should have absolute confidence on your preparation. Just go with enthusiasm, happiness into the exam. And by the way, time to apply will come shortly. All of you have your documents ready? All of you have your previous marks cards, your Aadhaar card, whatever is required. Keep it handy. When I spoke to many of you, some of you did not attempt the exam when I took interviews. I don't know whether somebody is here or not. When I took the interview, some of the students did not attempt the exam because they did not have the Aadhaar card to apply. Alright, so keep those ready. We will have to apply shortly. Notification will come shortly. Alright, okay. In a population of 1,000 individuals, 360 belong to genotype AA capital, 480 to capital A small a, and 160 to small a. Based on this data, the frequency of allele A in the small a in the population is. Come on, you should instantly tell this. What is given? Total individuals. Always Hardy-Weinberg equation. Write down. Total individuals. What is the total number of individuals? 1,000. What else is given? P square is 360. 2PQ is 480. Q square is 160. Correct? 160. Now, what do you have to find out? Frequency of allele A, small a, is, what is it that you have to find out? Q. You have to find out Q. Q square is given, which is 160. What is Q? Is it square root of 160? Square root of 160 by 1000. You have to bring it down to 1. Square root of 160 by 1000. Correct? So that is square root of 16 by 100. Square root of 16 is 4. Divided by square root of 100 is 10. 4 divided by 10 is 0.4. 4. 0.4. You can do it that otherwise also. 160 divided by 1000 is 0.16. Square root of 0.16 is 0.4. If you get confused with square root of decimals, keep it whole numbers only. Square root of 16 divided by square root of 100. Square root of 16 is 4, square root of 100 is 10, 4 divided by 10 is 0.4. That's all. Simple. You should feel very happy if you get questions related to genetics, Hardy-Weinberg principle, ratios. Next. Did all of you get it? Okay, now tell me, if I ask you, in the same population, how many dominant alleles will be found in the gene pool? How many dominant alleles are found in the gene pool? How many dominant alleles are present in the gene pool? How? How many dominant alleles are there? What is the total number of alleles present in the gene pool of this population? Huh? What is the total number of alleles present in the gene pool of this population for that particular gene? 1000, huh? How many is the total number of alleles for this gene in the gene pool of this population? How many people are there? 1000. How many alleles do each one of them have for this gene? 2 alleles. So what is the total number of alleles in this population for that particular gene? 2000. Of that, how many are dominant alleles? How many are homozygous dominant? 360 are homozygous dominant. Each one of them are having 2 dominant alleles. So among the dominant individuals, you have 360 into 2, 720 dominant alleles. How many are heterozygous? 480. So how many dominant alleles are there among them? 480. Total number of dominant alleles is 720 plus 480, which is 1200. That also can be the question, no? Common sense. There is nothing to remember, or there is no advanced logic to apply here. Common sense. There are 1000 people. Totally for this gene there are 2000 alleles. How many of those alleles are dominant alleles? All the data is given. 360 people are homozygous dominant. So each one of them have 2 dominant alleles. 480 people are heterozygous. So each one of them have 1 dominant allele. Rest of them are homozygous recessive. So count the number of alleles, simple arithmetic. Okay. A human female with Turner syndrome has 45 chromosomes with XO, has one additional X chromosome, exhibits male characters, is able to produce children with a normal husband. What is correct? 45 chromosomes with XO. Turner syndrome is monosomy. Or happening because of what? Non-disjunction of X chromosome. Non-disjunction of X chromosome. Presence of additional X chromosome is what? What condition? If it is a male, it is Klinefelter syndrome. If it is a female, is there a possibility that a female can have an additional X chromosome? Non-disjunction of X chromosomes in the mother will lead to the gamete, a ovum having 2 X chromosomes, fertilized by a sperm having X chromosome. Possible, no? Even for Klinefelter syndrome, the 2 X chromosomes come from the mother only. Non-disjunction of X chromosomes in the mother only. Sperm has Y chromosome, so it becomes Klinefelter. Otherwise, 3 X chromosomes. What will it be? What is it called? Super female. Nothing super about the female. She will have reproductive suppression, no reproductive development. But because of one extra X chromosome, it is referred to as super female. So in movies and all, if somebody refers to some women as super female, you should ask, is it because of her behavioral traits or is it genetic? Is she genetically a super female? All right. Select the correct option. Direction of RNA synthesis, direction of reading the template DNA strand. Direction of RNA synthesis will be in what direction? What direction? 5 to 3. Anything is 5 to 3. Any synthesis is 5 to 3. Any DNA or RNA production happens in 5 to 3 direction. Replication 5 to 3. So the template on which this 5 to 3 direction RNA is being produced should be in the direction 3 to 5 because it is complementary. 3 to 5. Template is 3 to 5. Synthesis is 5 to 3. You understand? What is the direction of the leading strand of DNA during DNA replication? Leading strand of DNA during replication has polarity dash. 3 to 5. Why? Whatever is produced, whatever is replicated as leading strand as the template will have continuous replication. The one which is produced with lagging strand as the template has interrupted replication or discontinuous. That is why what is the replication of DNA referred to as? It is semi-conservative because one of the strands of the parent DNA is a part of the daughter DNA and it is semi-discontinuous. Why semi-discontinuous? One of the strand is continuously replicated, the other strand is discontinuously replicated. That is why it is semi-discontinuous. Got it? Done. Commonly used vectors for human genome sequencing are human genome sequencing. What do we use for human genome sequencing? tDNA, BAC, YAC, expression vectors, TA cloning vectors. What do we use? Human genome sequencing. Human genomes are bigger genomes or smaller genomes? Bigger genes or smaller genes? Human genes are big. They are longer in their nucleotide sequence span. So for longer DNA fragments to be replicated or sequenced, by integrating it with some vector, what vectors do you want to use? BAC and YAC, bacterial artificial chromosome and yeast artificial chromosome. We cannot use plasmids. So in our human genome project, BAC and YAC were used extensively. Forelimbs of cats, lizards used in walking, forelimbs of whales used in swimming, and forelimbs of bats used in flying are an example of what organs? Homologous organs. What kind of evolution? Divergent evolution. Can we call it adaptive radiation? Can we call it adaptive radiation? Homologous organs, fine. Adaptive radiation, also there will be variations in the organs because of different environmental exposure. So can we call it adaptive radiation? Can we call all of this as adaptive radiation? Why not? What is adaptive radiation then? We have discussed this again, previous paper only. See the continuity, the same things get modified into something else, that's all. As we go from paper to paper, you will see it will all be the same things and related things. What else can you give? Correct, no? You cannot ask who was the second wife of Francis Crick. You have to ask the relevant questions only. All of that has been already dealt with. Already. Huh? Why not adaptive radiation? Adaptive radiation is not related to different species. Adaptive radiation is the variations in the morphology that you find in the organisms belonging to the same species because of different environmental exposure. That is adaptive radiation. The same species changing in different, displaying variations because of their environment, That is adaptive radiation. Our color, our body type. Like we are more brownish in color because we are a temperate country. We are a temperate country exposed to sunlight, our pigmentation is more. The tropical countries, Europeans are more, they are less colored. We are more colored. Alright? We don't say, color is the star there, not absence of color. So it should be reverse. Europeans should use tanning creams, not Indians using fairness creams. Alright? Because tanning is protective. Lack of melanin is damaging. Have you seen those? You eat banana in the morning, no? Banana. You saw the skin of banana? Do you find that kind of skin in Indians? You will find that kind of skin in a lot of Europeans. Spotted pigmentation, like spotted deer. We have uniform pigmentation. Alright. So who is more colorful? We are more colorful. Just because they made us believe that they are a superior race and they could not make everybody believe. But some of us believed that they are a superior race and fair is beautiful. There is a statement which I like very much. I came across that statement long back, maybe when I was in MBBS time. If there is light in the soul, there is beauty in the person. The best definition of beauty that I read. Somebody who is happy, somebody who is a good person by nature, that person is more beautiful. Not based on the height, weight, color, texture. No. Which one of the following are analogous structures? Wings of bat and wings of pigeon. Are they analogous? Homologous. Because both of them have the four-limb structure as the foundation of it. Gills of prawn and lungs of man. Are they analogous? Gills of prawn, do they have the similar structure as the lungs? But gills of prawn also helps in respiration. Lungs of man also helps in respiration. So are they analogous, both of them? They are analogous, they are not homologous. Thorns of bougainvillea and tendrils of cucurbita. They are also homologous. Thorns of bougainvillea helps in protection, tendrils help in climbing, support. Flippers of dolphin, legs of horse. They are also homologous. We'll finish off and then take the break. Alright, we have some 7-8 questions more, that's all. Which is the particular type of drug that is obtained from the plant whose one flowering branch is shown below? What is the plant that is shown? Huh? Datura, na? Look at it. Is it Datura? Is it not poppy? It is Datura? Okay. So what is the drug obtained from the Datura plant whose one flowering branch is shown below? Is Datura's flower responsible for its psychotropic effect? Or what is responsible in Datura? Datura seeds. Datura seeds cause hallucination. Datura fruit and seed. I don't think it is Datura. It is Datura? Okay, yeah. I'm not able to see the image properly here. Anyways, okay, what is Datura causing? Hallucination. It is a hallucinogen. At which stage of HIV infection does one usually show symptoms of AIDS? Within 15 days of sexual contact with an infected person, when the infected retrovirus enters host cells, when HIV damages a large number of helper T lymphocytes, when the viral DNA is produced by reverse transcriptase. When does the person show symptoms of AIDS? Option 3. When a large number of helper T cells are damaged. All right. Symptoms do not appear just suddenly. It is a constant battle. It is a constant battle. When the virus becomes stronger than the immune system, then the symptom starts appearing. Based on the damage to the immune system, the symptom starts becoming more and more intense. AIDS is the final stage, acquired immunodeficiency syndrome. AIDS is the final stage of it. Initially, it will be unexplained fever and weight loss. Unexplained fever and weight loss. Then it will move on to becoming lymph node enlargement, generalized lymph node enlargement, reflecting that there is some inflammation and infection going on throughout the body. Then it will be AIDS-like symptoms. AIDS-like symptoms. Some opportunistic infections will start catching up. Then it is full-blown AIDS. Full-blown AIDS where there is a significant suppression of the immune system leading to multiple diseases of multiple organs and systems also. To obtain virus-free healthy plants from a diseased one by tissue culture technique, which part of the diseased plant will be taken? What part do you want to take? Apical meristem only, palisade parenchyma, both apical and axillary meristem, epidermis only. Option 3. Meristems are unaffected by infections. Meristems are the healthiest tissue that you can find in the plant body. Parenchyma, epidermis, all of that can be infected, but meristems don't catch infection. So you can take either the axillary apical meristem, axillary meristem, either of them you can take. What gases are produced in anaerobic sludge digesters? Methane and carbon dioxide only. Methane, hydrogen sulfide and carbon dioxide. Methane, hydrogen sulfide and oxygen. Hydrogen sulfide and carbon dioxide. Methane and carbon dioxide certainly. Oxygen definitely not, because it is already mentioned as anaerobic sludge digesters. Now we have to decide whether you have hydrogen sulfide also or not, between 1 and 2. Hydrogen sulfide is also a part of gas produced in the anaerobic sludge digesters. Nothing much to explain here, we will just keep moving forward. Just as a person moving from Delhi to Shimla to escape the heat for the duration of hot summer, thousands of migratory birds from Siberia and other extremely cold northern regions move to Western Ghats, Meghalaya, Corbett National Park, Kiladio National Park. Where do they move? Kiladio National Park. Where is it? Rajasthan. It is a UNESCO World Heritage Site. So very famous for migratory birds. What? Siberian crane. Siberian crane. A lot of cranes also migrate to a place near my native village. 5 kilometers from our village is a place. It is named after, the village is named after cranes. The village name is Belluru. Belluru. Cranes are referred to as Kokkare in Kannada. So the village name is Kokkare Belluru. Very popular destination. During season you will find a lot of birds, a lot of cranes. It has a lake there. Every house, the trees in front of every house will have thousands of cranes. The villagers take care of those cranes. They don't damage those cranes or their nests. It's like guests. They come during the season and they go. They are also migratory birds. Western Ghats and Meghalaya are all biodiversity hotspots. A lot of biodiversity is found in Western Ghats also, and even in the northeastern region, Meghalaya. Corbett National Park. What is it popular for? Jim Corbett National Park, tiger. It is a tiger reserve. Given below is a simplified model of phosphorus cycling in a terrestrial ecosystem with 4 blanks. Identify the blanks. A, B, C, D. A, B, C, D. What are those? A is, where is it coming from? Phosphorus cycle begins with what? Primary source of phosphorus? Rock. Then where does it? B is rock minerals from which things are coming. C is producers from whom consumers are taking things. Yes. D is litter fall. Okay, C is producer, no? C is producer from whom consumers are getting the phosphorus. From soil solution, producers are taking it up. That is C again. So C is at the center of all of this. C is at the center of all of this, and A is detritus. B is rock. All right. Given below is the representation of the extent of global diversity of invertebrates. What groups do the four portions represent respectively? A, B, C, D. A is insects. Clear. After insect, who is the next one? More in diversity than the lesser in diversity than the insect, but second in number. Molluscans. Then crustaceans. Then other animal groups. All right. A scrubber in the exhaust of a chemical industrial plant removes what? What does the scrubber remove? Gases like sulfur dioxide, particulate matter of the size 5 micrometers or above, gases like ozone and methane, particulate matter of the size 2.5 micrometers or less. Scrubbers are not particulate filters. They are reactors. They react with sulfur dioxide gas and remove them. How do they do it? How do they do it? Scrubbers use water or some chemical solutions which can neutralize these gases. All right. If sulfur is not removed, sulfur dioxide is not removed from the exhaust or the industrial fumes, what will happen? Sulfur dioxide goes and combines with moisture in the air and becomes what? Acidic, sulfuric acid. So it will cause acid rain. So removing sulfur dioxide is important from that. If 20 joule of energy is trapped at the producer level, how much energy will be available to the peacock as food in the following chain? At the producer level, that is plant has 20 joules, mice will get 2 joules, snake will get 0.2 joules, peacock will get 0.02 joules. What is this law? Bioenergetic law or 10% law, or who gave this? Is it Haeckel's law? Haeckel's law is what? Haeckel's law is recapitulation theory, no? Haeckel's law is recapitulation theory, which is phylogeny mimics ontogeny. Ontogeny repeats phylogeny. Correct. So the life cycle repeats evolutionary cycle. All right. Done paper 2. We'll start with paper 3. Okay. Let us see the continuity also in this. 1, 2, 3, 2013, 14 over 2015. Which one of the following matches is correct? Phytophthora, aseptate mycelium, Basidiomycetes. Phytophthora is Oomycetes. Oomycetes have what type of mycelium? See, all of these are fungi. Phytophthora, Alternaria, Mucor, Agaricus. All of these are fungi. Characteristic structural feature of fungi is the presence of hyphae. Hyphae are like branches of the fungi. Hyphae. Now these hyphae might have septa, partitions, or fused. The entire hyphae might be uniform without any septum. Septaless hyphae without septa are called what hyphae? Cenocytic hyphae. Fused hyphae. If they have septa, it is called septate. Oomycetes, there is no septum. Hyphae is aseptate. Cenocytic. There are 4 classes of fungi that we study. What are those 4? Oomycetes, Basidiomycetes, Zygomycetes, Basidiomycetes and Deuteromycetes. These are the 4 classes of fungi that you have to remember. Oomycetes, Zygomycetes, Basidiomycetes and Deuteromycetes. Most of the fungi that we eat, mushrooms, they are all Basidiomycetes, Basidium. Agaricus is Basidiomycetes. Basidium is the fruiting body there. Alright? Okay. So Phytophthora is Oomycetes, not Basidiomycetes. So it is aseptate mycelium is correct, cenocytic mycelium or aseptate mycelium, but it is not Basidiomycetes, Oomycetes. Alternaria, sexual reproduction absent, Deuteromycetes. That is correct. See, the speciality of Deuteromycetes is in all the other three classes of fungi, both sexual and asexual reproduction is seen. But Deuteromycetes is a class of fungi where sexual reproduction is absent. They don't have any sexual reproduction. Irrespective of what physical characteristics they display, if a particular fungus does not show sexual reproduction, then it is put in the Deuteromycetes. Alright? Mucor, reproduction by conjugation, and it's an Ascomycetes is the question. Mucor is Zygomycetes, not Ascomycetes. Mucor is Zygomycetes, reproduces by conjugation is correct, but it is not Ascomycetes. Agaricus, parasitic fungus, Basidiomycetes. Agaricus is a saprophytic fungus, not parasitic. Most of the fungi are saprophytic. You don't find parasitic fungi. Agaricus belongs to Basidiomycetes. The edible mushroom that we eat is Agaricus. Read the following 5 statements and select the option with all correct statements. So there are 5 statements. Options are combinations of statements to be correct. In such questions, what do you have to do? Catch hold of one which you are absolutely sure. Tell me one statement which you are absolutely sure that it is correct. For A is absolutely correct because it is related to succession, and these are the primary, these are responsible for primary succession. These are the, what do you call those, the one which will initiate succession? What species? Pioneer species. They are the pioneer species. Mosses and lichens are the first organisms to colonize a bare rock. A is definitely correct. So an option without A should be ruled out. Which options do you rule out? 2 and 4. Now between 1 and 3, you see A, D are common. So statement D is also correct. Main plant body in bryophytes is gametophytic, whereas in pteridophytes it is sporophytic. We also validate that. That is correct. D is right. Now the question is whether C is wrong or E is wrong. Or the other way is C is correct or E is correct. That will tell you what is right. If you are able to conclude, excellent. Otherwise, 50-50. You are already eliminated. You already eliminated 2 options. Between the 2 options, you have to make a judgment call whether you have to go with one by your instinct or leave it. It is like investment. If you have more risk appetite, you will invest. You will invest in high return, high risk investments. If you have less risk appetite, you will play safe. Let us look at C and E. C. Coralloid roots in cycas have VAM. What is VAM? Vesicular, arbuscular, mycorrhiza. Mycorrhiza are symbiotic what? Fungus. Mycorrhiza are symbiotic fungus present in the roots, coralloid roots. But coralloid roots in cycas, what do they have? They don't have mycorrhiza. They don't have a mycorrhizal association. They have cyanobacteria association. Nitrogen fixing cyanobacteria are present in the coralloid roots of cycas. We have seen this in one of the previous papers, the last 2 papers that we discussed. Look at the continuity, the same things altered. Okay, coralloid roots in cycas have cyanobacteria association, not VAM. So, statement C is wrong. That means option 1 is wrong. Option 3 is right. Check for E. In option 3, you have one more as E. In gymnosperms, male and female gametophytes are present within sporangia located on the sporophyte. True or false? Gymnosperms, gametophytic generation is dominant, sporophytic generation is dependent. No, in bryophytes that is. In gymnosperms it is reverse. Sporophyte is dominant and gametophyte is dependent. Male and female gametophytes are present within sporangia. What sporangia? There are two types of sporangia. Microsporangium and megasporangium. That is correct. They are present on the sporophyte. E statement E is right. So A, D, E. Selaginella is a homosporous pteridophyte. Selaginella is not homosporous. Selaginella is heterosporous. Again, Selaginella we have seen in the previous two papers also. Okay. What do you mean by heterosporous? There we have seen it as oogamous, oogamy. Heterospory means both the gametes are different. They are not similar. So Selaginella is oogamous. That is what we have seen. In which of the following gametophyte is not independent and free living? Gametophyte is not independent and free living in which division? Pteridophyte. Pteridophyte. Or even gymnosperms also, if you look at it, gymnosperm, angiosperm, all of that, gametophyte is not free living. Neither it is independent. So look at the options. Funaria, Marchantia, Pteris. Funaria and Marchantia are bryophytes. In Pteris, Prothalus is free living. It's a pteridophyte, but Prothalus is free living, though it is not long lived. Option 4 is Pinus. Pinus is pine tree, which is gymnosperm. We know for sure, even if you are not sure of the other three. Gymnosperm, certainly gametophyte is dependent on the sporophyte. Not at all free living, definitely not free living. So you can go with option 4. Which one of the following statements is wrong? Algin and carrageen are products of algae. Is it right? It is right. These are all directly out of NCERT. Algin from brown algae, carrageen from red algae. Carrageenin from red algae. Agar is obtained from Gelidium and Gracilaria. True or false? True. Gelidium and Gracilaria both are red algae. Chlorella and spirulina are used as space food. Yes. Yesterday only we discussed about chlorella and spirulina both. Chlorella and spirulina are used as space food because of their high nutritive value, easily absorbable proteins. Mannitol is stored food in Rhodophyceae. Rhodophyceae is red algae. But mannitol is stored food in Phaeophyceae, brown algae, not in Rhodophyceae or red algae. The guts of cow and buffalo possess Fucus species, Chlorella species, methanogens, cyanobacteria. Fucus and Chlorella are algae. They are not present in the gut of cows. They are algae, they are photosynthetic, they are present outside. Cyanobacteria involved in nitrogen fixation, they are not associated with gut of cow. Cows are one of the major contributors for greenhouse effect, global warming, methane gas. The gut of cow has bacteria which can produce methane gas. The bacteria are methanogens. All right. So another problem, a large part of the world, meat eaters eat beef. Beef is rich in protein. Protein content of beef is high, and beef is cheaper in terms of its price. Meat is more, cost is less. So lot of people eat beef across the globe. Beef eaters grow these cattle, nurture these cattle for the purpose of eating. Otherwise, there will not be so many cows. Unless other than for eating, if you don't eat cows, there is not so many cows available in the on the earth. To eat, we are cultivating so many animals, and the animals are causing food crisis. Animals are causing global warming. All of that for us to eat. Cows, major contributors of global warming. And also cows are water guzzlers. They drink a lot of water. So cows consume a lot of water, cows consume a lot of grains, food, they produce methane. Problem for the globe, global warming. All right. Male gametes are flagellated in, see again, flagellated gametes, non-flagellated gametes, isogametes, anisogametes. Continuously for 3 years the same question has come. Male gametes are flagellated in Polycyphonia, non-motile gametes, Anabina, asexual reproduction. The Anabina is a cyanobacteria which is asexual reproduction. Spirogyra, chlorophyte, reproduces by conjugation, no flagellated gametes. In one of our previous papers we have seen Ectocarpus, brown algae. Ectocarpus, male gametes are flagellated and motile. Vascular bundles in monocotyledons are considered closed because vascular bundles in monocotyledons are closed. Why? That means if the vascular bundles are closed, what is the implication? What cannot the plant do? Secondary growth. Secondary growth is not possible if the vascular bundles are closed. Why is it closed? A bundle sheath surrounds each bundle. Cambium is absent. There are no vessels with perforations. Xylem is surrounded all around by phloem. Why do you consider the bundles to be closed is the question. Of the 4 options, why do you consider the bundles to be closed? Or when do you consider the bundles are closed? When they don't have cambium. The other options are not related to secondary growth. What is bundle sheath? Is the covering around the vascular bundles, and there is something under the epidermis of the monocots. What is it? At the outer end of the cortex. Sclerenchymatous hypodermis is present in monocots. That is what makes them hard. You pull the leaves of this angiosperm plants, dicotyledonous plants. You can pull off the leaves. Not much damage will happen to your hands. But if you just try to drag the grass and the grass goes through your hand, what will happen? It will cause cuts. Why? Sclerenchymatous hypodermis is present in monocots. The floral formula given is for, where is the floral formula? Is there a floral formula in the paper? Okay. Okay. So what is represented? Actinomorphic symmetry, bisexuality, presence of tepals, superior ovary. Is that what is given? What is the floral formula of Allium, Sesbania, Petunia, Brassica? No. I can't conclude on this. Okay, we'll skip this. A major characteristic of the monocot root is the presence of open vascular bundles. Is it? Are the vascular bundles open in monocots? No. Scattered vascular bundles. Scattered vascular bundles. It is a characteristic of monocots, but not of roots. It is of stem. Vasculature without cambium. Same question we saw in the previous question. Same thing we saw in the previous question. Closed vascular bundles are found in monocots. So vasculature without cambium is correct. Cambium sandwiched between phloem and xylem along the radius. That is seen in dicot roots. Dicot roots, not in monocots. Keel is the characteristic feature of flower of what? Tulip, indigofera, aloe, tomato. Keel flower. Keel, a structure formed by the fusion of 2 anterior petals. Keel petal is what type of estivation? Vexillary estivation. Vexillary estivation is seen in which family? Fabaceae. Fabaceae. Now the question is, which of these belongs to Fabaceae? Tulips belongs to the lily family, Liliaceae. Aloe belongs to the lily family again, no keel. Tomato, Solanaceae. So Indigofera is Fabaceae. Just a moment. I'll just open up something else also. Okay. All right. Next question. Perigynous flowers are found in guava, cucumber, China rose, rose. What is perigynous flowers? Half inferior or half superior. That means the gynoecium and all the other whorls are at the same level. So guava and cucumber, epigynous flowers. China rose, we have seen this. China rose, we have seen it in the previous paper. China rose has hypogynous flowers. Rose is perigynous flowers. Leaves become modified into spines in opuntia, pea, onion, silk, cotton. Opuntia. Opuntia is a xerophytic plant. Transformation or modification of leaves into spines is for the prevention of transpiration loss of water. What kind of photosynthesis do you see in xerophytic plants? A special type of photosynthesis? Crassulacean acid metabolism. CAM photosynthesis. Crassulacean acid metabolism. All right. What is special there in CAM photosynthesis? Stomata opens during the night time, absorbs, takes in carbon dioxide, converts it into an acidic form, crassulacean acid, and carbon dioxide is re-released into the chloroplast during the daytime when the sunlight is available for photosynthesis. So interrupted photosynthesis. It is not in continuity. For all other photosynthetic plants, other than the xerophytic plants with CAM photosynthesis adaptation, when there is sunlight, there is light reaction as well as dark reaction happening simultaneously. Light dependent and light independent reaction. So light independent reaction also happens in the presence of sunlight only, during daytime only, for all other plants. But these fellows have mastered the art of fixing carbon dioxide during the night time and using it during the daytime. There also, even in CAM photosynthesis plants, light dependent reaction and light independent reaction, both of them are happening during the daytime only. Understand that? Night time, it is only carbon dioxide that is being taken in and converted into a storage form. But both reactions are happening during daytime only, not during night time. Should be clear with that. The structures that are formed by stacking of organized flattened membranous sacs in the chloroplasts are. This is easy. What do you call the stacks? Each of the plate in the stack? Thylakoids. The entire stack is called grana. Where do you find cristae? Cristae? Mitochondria. Which part of mitochondria? Inner membrane of mitochondria. Cristae are the infoldings of the inner membrane of mitochondria. Grana, we saw that. What is stromal lamellae? Stromal lamellae is what? Is that also thylakoid? Stromal lamellae is also thylakoid? Structurally, is it similar to thylakoids? Stromal lamellae? No? Stromal lamellae is the connecting membrane between adjacent grana. They connect the grana. Structurally, they are similar to thylakoids, but they are not stacked up to be a part of the grana. They are connectors of grana. Stroma, where do you find? In the chloroplast, stroma. What is equivalent to stroma in mitochondria? If stroma is to chloroplast, dash is to mitochondria. Matrix. Chromosomes in which centromere is situated close to one end are called metacentric, acrocentric, telocentric, submetacentric. Acrocentric. What is metacentric chromosome? Centromere is at the center. Centromere is at the center of the chromosome, that is metacentric. Acrocentric is? See, okay, we will just, at the center is metacentric. At the tip is, at one end is telocentric. Close to the tip but not at the tip. Acrocentric. Close to the center but not at the center. Submetacentric. That's all. There is central centromere, terminal centromere. Close to the terminal but not the terminal one. Close to the center but not the center one. So you have to remember only two things here. Metacentric, telocentric. Close to it is acrocentric. Close to the center is submetacentric. Select the correct matching in the following pairs. Smooth endoplasmic reticulum. Oxidation of phospholipids. Is it correct? What is, if you want to correct the statement? They can produce it. Smooth endoplasmic reticulum can synthesize phospholipids. They don't break down phospholipids. Lipid synthesis, detoxification is done in smooth endoplasmic reticulum. All right. Carbohydrate metabolism it contributes to. Smooth endoplasmic reticulum, synthesis of lipids. Is it correct? Correct. Rough endoplasmic reticulum, synthesis of glycogen. Wrong. Rough endoplasmic reticulum's role is to contribute in the synthesis of proteins. Protein synthesis. Rough endoplasmic reticulum, oxidation of fatty acids. Wrong again. True nucleus is absent in. Among the four options, we have to identify which one is a prokaryote. We have to identify which one is a prokaryote. So, true nucleus is absent in Anabaena. Is Anabaena a prokaryote? Yes, it's a bacteria. Mucor. Mucor is fungus. Vaucheria. Again, it's a eukaryote. Volvox. Volvox is algae. So all the other three are eukaryotes. Anabaena is a blue-green algae, cyanobacterium, devoid of true nucleus. Which one of the following is not an inclusion body found in prokaryotes? What are inclusion bodies? Non-membrane bound masses that you find inside the cells. It can be crystals, it can be lipid mass, it can be phosphate mass, calcium masses, some organic compounds in the form of a mass. Okay, non-membrane bound structures that you find. Which one of the following is not an inclusion body? Phosphate granule, can it be an inclusion body? Yes, phosphate granule can be inclusion body. Synophician granule, nitrogen storing masses. These are nitrogen storing masses. Glycogen granule, they are also inclusion bodies. Polysome is not inclusion body. What is polysome? Multiple ribosomes bound to a single mRNA. That is a polysome. There is one mRNA, let's say at one end, at the beginning, translation process has started and one ribosome is at a certain distance from the initiation point. Now one more has started and it is also translating. Multiple ribosomes attached to a single mRNA for protein synthesis. That is a polysome. Transpiration and root pressure causes water to rise in plants by. This also we have discussed already. Transpiration causes what? Pull. Root pressure causes. Phototropism is the movement of plant towards the light. Auxin accumulates in the opposite side, where the plant is opposite side of the light exposure, so that plant tilts towards the light exposure. What are the other tropic movements you can remember? Phototropism is one, then geotropism, then hydrotropism. Three important tropic movements. All right. In a ring-girdled plant, The shoot dies first, the root dies first, the shoot and root die together, neither root nor shoot will die. In a ring girdled plant, what do you mean by a ring girdling of ring? What do you mean by ring girdling of plant? What do you mean by ring girdling of plant? See, in the shoot, ring girdling of plant is you are cutting the shoot to a certain depth, like a ring on all sides of the plant. What do you cut? See, when you are cutting, when you are going from outside to inside, you are chopping off layers of the trunk. What are you cutting off from outside to inside? As you move from outside to inside, what are you removing first? Bark. Let's assume it is not a secondary growth plant. You are cutting off epidermis. Then you are cutting off layers of the cortex. Then you are cutting off the endodermis. Then as you move inwards further, what do you encounter? Vascular bundles. In the vascular bundles, what do you get first? Phloem or xylem? What is towards the outside? Phloem or xylem? Phloem is towards the outside. You cut off the phloem. That's all. What you cut? You would take a ring. You cut it like a ring. From epidermis to phloem, you chop off all around the plant. You are cutting till the phloem. That is ring girdling of plant. When you do this, what will die first is the question. Shoot or root? The part of the plant above that or the part of the plant below that? Think logically. Why?
Because that food will not transport itself.
Water transportation is still happening. Mineral transportation is still happening via xylem from the root to the shoot. So the shoot is able to survive. But the food that is produced in the shoot, that is leaves, will have to be transported to the root for roots to survive. Because you have cut off phloem by the ring girdling, food will not be able to cross below that. Will not be able to cross that and reach the lower levels, lower layers, or the lower part of the plant body. So plant body below the ring of girdling will die first than the upper body. Why will the upper part die then? Why will the shoot die? It is anyways getting water, no? If root dies, how will the water reach the shoot? Alright? If root dies, water will not reach the shoot. Typical growth curve in plants is sigmoid, linear, step-shaped, parabolic. Sigmoid. Any growth is generally sigmoid. Initial growth will be slow, then there will be a significantly rapid growth till it attains its optimum growth, and then it stabilizes, and if the conditions favor, it will continue to remain stable. If conditions become worse, it starts declining. Then it becomes if you look at the lifespan of growth, it will become a bell-shaped curve. Growth, increase in growth, stable, continuing the mass stably, and decline in the mass that is there in the body. From childhood to death, if you look at death of a person when he reaches ripe old age, see, the mass keeps going down. It will not become a bell-shaped curve. The elderly man at 100 years of age will not become as much as what he was when he was born, but it will be a declining curve. Sigmoid curve is the growth curve. Which one gives the most valid and recent explanation for stomatal movements? Why does the stomatal movement happen? What is the recent explanation? Transpiration, potassium influx and efflux, starch hydrolysis, guard cell photosynthesis. See, transpiration and photosynthesis contribute indirectly for the opening and closing of stomata. Transpiration will lead to loss of water. Transpiration will lead to loss of water from the stomata. Stomatal cells also, making the stomatal cells become flaccid. Flaccidity of the stomatal cells will close the stomata. Guard cells will close the stomata or open the stomata. If the guard cells become flaccid, they lose water. will the stomata close or open? Close. Transpiration is indirectly influencing stomatal closure. Transpiration brings about stomatal closure. Photosynthesis in the guard cells, how will it contribute to stomatal opening and closure? It is not the primary influence, primary effect, primary cause, but how does it contribute? Photosynthesis leads to the production of carbohydrates, sugars, simple sugars, glucose, fructose, these are the things that are produced. They are osmotic substances. Osmotic substances increase in the concentration of osmotic substances within the guard cell. What will it do to water? What will it increase? It increases the osmotic pressure. So, guard cells will draw water from the surrounding xylem vessels, becoming turgid. Turgidity of the guard cells will open the stomata. So, transpiration and photosynthesis both contribute to stomatal opening. Transpiration contributes to closing of stomata. Photosynthesis in the guard cells contribute to opening of stomata. They are not the primary effects though. Starch hydrolysis is also one of the theories, earlier considered theories, where starch is hydrolyzed into simple sugars, which will lead to increased osmotic pressure, causing water influx, increasing the turgidity, opening of guard cells. That was the earlier theory. Now the accepted theory is active transport of potassium ions into and outside of the guard cells. So when potassium ions, when the guard cells have to open, potassium is transported actively into the guard cell, increasing the osmotic pressure. More water is drawn, turgor pressure increases. The guard cell becomes more rigid, turgid, opening of the stomata. So influx and efflux of potassium being done actively is the most accepted theory at this point of time. All the other three are also associated with opening and closing of stomata, but their influence is lesser compared to the influx and efflux of potassium as per what we agree to believe now. Hilum is a scar on the, where do you find hilum? Seed. What does it represent? What does the hilum represent? The site where funicle was attached when it was a ovule. When the seed was an ovule, the site of hilum is the site where funicle had attached. What is funicle? What is funicle? What is funicle to the seed in comparison to the embryo, the fetus? No, funicle is not the placenta. Placenta is placenta both. In plants also and in humans also it is placenta only. What is funicle? What does funicle do? Funicle connects the developing ovule to the placenta. What is an equivalent structure in humans? Umbilical cord. Umbilical cord, no? So funicle is like the umbilical cord of the ovule. Both of them are deriving nourishment from taking nourishment from the placenta and delivering it to the developing embryo. So hilum is a scar on the seed where funicle was attached, fruit where it was attached to pedicel, fruit where style was present, seed where micropyle was present. It's a seed where funicle was attached. Micropyle, where do you find the micropyle? What is micropyle? Micropyle is a small pore in the seed. Why do you need micropyle in the seed? What is the importance of micropyle? It helps in respiration. It helps in the entry of gases and entry of oxygen, exit of carbon dioxide and all that. But during germination, see what has led to seed dormancy? What has created dormancy in the seed? Why is the seed not germinating? Why is the seed not germinating until you put it in the soil and add moisture to that? The seed gets loaded with what? Abscisic acid. seed gets loaded with abscisic acid, which will prevent metabolism, which will prevent metabolic activity in the cells of the seed.